light pollution.
Yeah, it doesn't work. But yeah, so outside the city, I think you can. Especially the mountains are great. You know, the higher the altitude, the better, of course.
Okay, so that wraps up my announcements, and I would like to introduce Mike Jaqua. He's our presenter, and he is a science educator. And so, Mike, if you would like to maybe give a little background of yourself, and then you could start sharing your presentation.
Oh, background, got my degrees in microbiology, immunology with minors in education and cancer biology. I consider myself a biologist. Have about 20 years of research in academia and corporate, and about 10 plus years writing medical education materials. I also brief stint teaching biology graduate teaching assistants how to be better teachers. So
keep them busy.
yeah, so that's sort of like my background for this sort of stuff.
Great.
All my other degrees are in martial arts, so they don't count.
Okay, you can start sharing your screen if you'd like.
Okay. Got it. And go. Slideshow. There we are.
There.
There you go. CRISPR-Cas9. That's obviously sort of a, contrived model, although it's fairly accurate. CRISPR-Cas9, for those of you who haven't heard about the thing in the news or some such, is sort of the latest hot item for genetic engineering. It surpasses its predecessors, which I will talk about briefly later on. It's a pretty powerful tool, and impressed when I was how people discovered it, because I would have totally missed it. So I'm going to start out, and some of this will feel like it goes back and forth a bit. It's because explaining this is hard to do in a cohesively straightforward manner. So you'll hear some information more than once. All right, so starting off with a quick review of DNA and genes. I know a lot of us had it in high school and may have forgotten it since then. So we're going to start out working from the cell out from the nucleus. That's the cell and its nucleus. Most of you have seen chromosomes, and that's just like packed genetic material. And for those who don't remember, there's 23 of them, one of which is an X or a Y or both, depending on who and what you are. This is the, this is sort of the artist's rendition of unwinding the DNA from the chromosome. And the genes are found in this part of the double helix. That's what they call this twisty shape of DNA. And then you'll see this more later. Things that DNA are made up of are these nucleic acids, or as they're sometimes called, bases. And hopefully it just came over there and showed you what they are, where they are. So each one of those little posts is like a nucleic acid. Now we'll go quickly to DNA and RNA. Most of you have heard about it. DNA has all the genetic information encoded on its genes. It's in there all the time, till and unless it's time to use one. Oh, sorry, I'll get to that next. It's a double helix. Difference is that RNA is single-stranded rather than double. That's so that it can be read easily. And there are your 4 nucleic acids, A, C, G, and T. See those from time to time. So remember ACGT, that's your highway to the cell. And let's see, okay. So this is, if you need to use DNA, you're going to transcribe it. It's like scribes, they write from one thing to the next. Transcribe it to RNA. You do what's called translation to proteins represented by those beads at the end. See these codons, there are 3 nucleotides or nucleic acid bases, and that's like the unit that you encode proteins in. It's kind of important because it has to do with where and how the CRISPR will bind. So you'll kind of see this not directly referred to, but indirectly. It's that chain of amino acids made up of amino acids rather than nucleic acids. Don't think of. And then the specific sequence of that DNA determines the protein that will be made. And if you mutate it, like the RNA I'm showing here, you can change the entire protein. And that's all you need is just one base to do that. You don't have to change 3 like a codon. Okay, what's CRISPR-Cas? Should have been 9, but that's okay. First thing, what they stand for, and you'll see this later, clustered regularly interspaced short palindromic repeats. That's why they call it CRISPR instead of all that. Cas is the CRISPR-associated system. And this is what they're made of. The CRISPR is made of RNA. It's a strip of nucleic acids, or RNA, not the DNA. And the Cas is a protein. The RNA guides the DNA to, it guides the RNA of the CRISPR to a specific portion of DNA. Then it cuts that. And you'll find out a couple of reasons why that cut is sort of the most important thing it does. Okay, here's what that long name stands for. Clustered regularly interspaced. See these spacers in between, those are viral sequences. And we'll get to that a bit again later. And then within those things in between the spacers, that's where you find what they call the palindromic repeats. And now I've sort of bracketed where they are. This parts that's blue, if you flip it, match to the part that's red. And if you look at it, they're not letter matches, they're what we call complementary matches. Each of these things bind to each other. So the A and the T always bind together, the C and the G always bind together. So those are complementary palindromic repeats. That's why I would have totally missed it if I'd been searching it. Okay, the important part of this is that, wow, a lot of stuff came in at once. See now the palindromic repeats, and when they fold over, when they fold over from the part of the gene they're in, they make this shape called a hairpin. And that's important for a number of reasons. In the case of CRISPR, it's part of what helps it to stabilize the structure. And I'm going to talk more about the gRNA later. That also includes the sequence that binds to the DNA. Again, you'll get more of what that means. So if you look at the little red twisty thing with white spikes sticking off of it, that's that hairpin loop. And it's kind of hard to see it, but it's in there and it's basically holding the protein together with the RNA. So it's sort of the stabilizing or joining structure. Then this is kind of that top thing turned to the other side. That little red strip is the RNA that helps it bind to specific DNA sequences. So just a brief going through the procedure. Got the called guide RNA and tracker RNA, and I always forget what tracker stands for. They're within the Cas protein, which is the bigger blue. they come in looking for that little 3 nucleotide sequence. So it's kind of like a codon that starts everything. Codons from earlier, that's the 3 base sequences. So that helps the CRISPR to grab hold of and read whatever DNA it matches to. Once that's all bound together, DNA it wants it, go again, cuts that DNA. Again, that's important. You'll see a lot about it as I go through. Now this is a brief video, as soon as it's willing to cooperate. See, I've got to get it back to...
Type of enzyme that can cut DNA. Within the viral genome, the Cas9 cuts the target DNA. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in...
From there, we'll see a little more about that later. I like that because it gives you a pictorial...
cultured cells, including...
Forward.
Stem...
Second here. Okay. There's what it does. So this is kind of gives you sort of a start as to what it does and a little bit of kind of how they found this crazy system. So its natural function is to act like an immune system for bacteria, which when I was in grad school, we had no idea why these things didn't get killed by bacterial viruses. Now we know, so things have changed a lot. What happens is if you get a viral infection, the CRISPR-Cas will bind to it. Well, okay, this is initially, it's one of the different Cas. You see Cas1 and 2. But it'll grab hold of a piece of the virus and cut it out, as those scissors indicate. And then that basically just destroys the viral genome. So just killed it. That's part of how bacteria keep from getting killed by viruses. And then what CRISPR will do is it'll keep some of the sequences. So if you see that little green section, that was cut out. The CRISPR will grab it, and it'll bring it down and incorporate it into the virus's own DNA or RNA. And that's important because later, see what all this is, it'll pull those spacers out if it gets infected by a virus. And if it's a virus it's seen before, one of those spacers will match it, and so it can react to it more quicker than it could the first time. So it's
Mike,
a memory.
can I interrupt for a second?
Yes.
So are you saying that this whole process was originally found to be a natural process in nature by bacteria stopping a viral infection?
It went a little bit different. They found it in bacteria. They found those palindromic repeats, and they were kind of like, why are these here? We see them all the time. And when they looked at the spacers and they sequenced them, they found that they matched a lot of viral sequences. So then they're thinking, okay, bacteria has bits of viral sequences in its DNA. What's it using that for? And it took them a little longer to find the CRISPR RNA binding to the Cas protein. Once they found that, they were able to see how it worked a little more, and they could actually throw in viral DNA and find that it did indeed bind to the complementary sequences of those spacers. So see, it was kind of a multi-step process. But once they figured out that those spacers match viral sequences, they knew that the bacteria was reacting to viruses in some way. And after that, it was a little easier to figure out what way it was doing. Just cutting up the viral DNA so that it can't do anything. It can't hurt the cell anymore. So it was a pretty complicated process, and in total took almost 10 years before they figured it out. So as mentioned, these protect against future infections by the same virus. It can react quickly, as opposed to the first time it sees the virus where it's a little slower to react. What I'm going to show you is how that compares to humans. You saw that the CRISPR takes pieces of the viral DNA, holds them in its genome, and then has a memory system that remembers that virus if it sees again. So in humans, we have the same sort of what we call adaptive immunity. It works via cells rather than small pieces of DNA, but it's very similar in how it works. So if we first get an infection, the immune system will activate some of its immune cells, go in, chew up bits of that infectious pathogen, and then it'll generate certain types of cells. One of them's the T cells. Everybody's familiar with antibodies. Those are made by something called B cells. The thing that people are not as familiar with is the T cells, and they are the ones that frequently have the job of killing viruses. They'll also kill things like cancer cells. So that's why I have this little, if you'll watch here, cell comes in, kills an infected cell. So if they find a cell with a virus in it, they basically just kill the whole cell. B cells have a slightly different function. The antibodies will bind to proteins as opposed to CRISPR that binds to DNA. Those can mark things for destruction. Then there's another part that's similar. Once the infection is cleared, some of the T and B cells become memory cells. Most of you know that if you see an infection a second time, your immune system will basically kick its ass really fast. This is why. This is also what vaccines generate. It'll generate the initial reaction, but then memory cells will stay, and that's why vaccines keep you immune over long periods of time. So the memory cells are there, just like the spacer sequences in CRISPR, to remember and react if it ever sees that same infectious organism again. Okay, now about how CRISPR was discovered, so a little more answer to Cheryl's question. Started way back in 1993 when they noticed those pieces of DNA and several species of virus that had these little complementary palindromic repeats. Realized at this point, for anybody who doesn't know or remember what a palindrome is, something that reads the same way forward and backwards. So like the word dad, you can read it either direction, it still says dad. Or if you want more complicated, the sentence that says, Abel was I ere I saw Elba. Napoleon's last words. So as you can see there, it took about 7 years to sort of figure out that these sequences were in all sorts of bacteria, start to understand that they belonged to viruses. First thing they figured out was that these repeats were actually transcribed. Remember, that's making something from DNA into RNA. From there, you'll get proteins. So then they found, this is what, 9 years later, they figured out what the Cas genes were that were near those CRISPR repeats. And I didn't really address this earlier, but the Cas genes are right near the CRISPR repeats in the gene, so it's easy to copy them all together. And here we are, 12 years after first discovering it, they have finally figured out that yes, it is the same as a lot of different viral DNA. Then they were able to show that the CRISPR sequences bound the viral DNA and cut it up at the sites where it binds. And so about that 2008, they finally figured out, oh, wow, bacteria have an immune system. Then they found out that they could actually pull that system out and work with it themselves and manipulate it. That was when things started to get really exciting because then they found out that they could just manipulate or change those little RNA sequences in CRISPR and generate something that would bind to any DNA sequence you wanted. And of course, it would cut it, so now you've cut up DNA. Took them all the way to 2015 before they first started editing human DNA. And that was one of the most important parts because then everybody went nuts. And since 2015, the number of publications on CRISPR has simply skyrocketed. Whereas prior to that, probably get familiar with all of them fairly quickly. I said I'd talk about the tools that preceded CRISPR and get an idea how things have developed. Restriction into nucleases, when I was a grad student, this was the only tool we had. And what you'd use is the restriction into nucleases or proteins, and they bind to specific bits of DNA. One of the limits to this, though, is you've got to find a protein that binds to the sequence you're interested in. And there are a limited number of those proteins, and they're extremely hard to engineer. You can't find one that binds the sequence you're interested in. So then they got a little better and discovered zinc finger nucleases. So they still cut the DNA, but it's a little easier to redesign the proteins and the sequences. Still time-consuming and expensive, but at least you could do it. Somebody came along with talons, which work pretty similarly, but it's a little easier to engineer them, and they were, so it was easier to make the batch to specific sequences. Then finally they found CRISPRs. These are they're easy to design, they're easy to make it bind whatever DNA sequence is because you only have to make a little short RNA sequence, everything else stays the same. And then it can cut any DNA site you want. It can also cut several of them simultaneously by just introducing, say if you want to cut 12 genes at the same time, engineer 12 strips of RNA and you put it into CRISPR-Cas and just put it in the cell and you cut 12 different sites at once. So extremely flexible and easier to work with. I'm going to talk just a little bit about something, and this is another discovery that was really important, was Cas9 is the one everybody's familiar with. Cas12a, or sometimes they call it CPF1, was something they finally discovered, and it's got some great benefits over Cas9 that have been growing in terms of their effect on genetic engineering. One of them is that CRISPR-Cas9 is a relatively large molecule. It's not super easy to deliver in cells, though you can do it. 12A is smaller, and it's actually really goes into cells without a heck of a lot of effort. So want to do your genetic engineering in cells, which you generally do, CRISPR's got some advantage there. Another thing is I've been showing you the RNA complex. It takes two RNAs, the gRNA and tracker RNA, both of which are required to cut. With 12A, it only needs a single strand of the RNA. So it's a little easier to engineer and work with when you're trying to make new ones to bind new sequences. Then there's another advantage to these. CRISPR-Cas9 does what are called blunt end cuts. That black line indicates that it just cuts along this same line when it makes its double-stranded break. Disadvantage to that is you end up with blunt ends on the DNA. They're just ends that were previously joined together. And it's hard to then insert new genes into that. It's doable, it's just hard. CRISPR, those black lines show, it cuts at two staggered sites. So you wind up with what they call sticky ends or complementary ends, and it's very easy then to make a matching or complementary sequence of DNA and just stick it in there. Another disadvantage is that CRISPR cuts really near that PAM site, which I showed you earlier. That's a 3-nucleotide sequence that initially binds to the target DNA and helps the rest of the RNA to lay out, grab hold of the entire cut site. But if you mess up that PAM sequence, then CRISPR is not going to be able to go bind more DNA. So you basically get one shot. With Casper, it cuts farther, with 12A, it cuts farther away from the PAM site. So even if you have trouble, or if it takes a long time to make the cuts and bind the new gene sequence, you get multiple chances to try it again. It also makes the 12A molecule persist a little longer. With Cas9A, once you've cut, that single protein RNA structure is kind of done with its job. Okay. This is a little more of the same video I had up earlier. Put it into a different place. It's supposed to start at 2:30, so let me.
Type of enzyme that can cut DNA. Living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations.
Okay, let's see if I can get this to move on this time. Nope, I can't.
And
Okay. Now, one thing I'd like you guys to know is a lot of what I'm presenting here is very technical. If you work as a biologist or had college education in it, this is relatively easy, probably somewhat easy to understand. But I know for a lot of you, this is, again, something you probably haven't heard since maybe high school. Please pipe in, because I realize that I have to go through this fast to get it done, but I also realize that a lot of this could easily just go right past you. I like that not to happen so much. So please do pipe in if some of this is puzzling and need it re-explained. So now I'm going to talk about applications for CRISPR. And you'll start to see just how powerful this tool is. What you can use it for on this list is you can activate or inactivate specific genes. So if you're activating genes, an example of it that's out there is that you can improve yeast. You primarily improve their protein output or their reproduction speed. These yeast produce biofuels. So here's CRISPR with a possible application of working on helping us generate new fuel sources as we try to get away from gas and oil. You can also inactivate genes, and one of the coolest applications of this I've seen is they're inactivating genes in mosquitoes so that they can no longer carry or transmit malaria. So they can do this, grow up a bunch of those mosquitoes, release them into the population, and try to get them to dominate the population and get rid of malaria in an area. Very powerful there. You can use it for what's called epigenomic editing. Epigenomic means outside of the genome. So these are things that affect DNA, but they're not part of the DNA sequence. So what you can do is you can take a CRISPR-Cas9 that is modified. It doesn't cut now. What it does is it'll carry that little epigenomic modulator. And that can cause it to either put methyl groups directly on the DNA, which basically clogs up that section of DNA so you can't read it. The other thing you see are the acetyl groups, which then grab the DNA and wrap it around those histone structures. Once they're wrapped around like that, they're inaccessible. And then also as it wraps around more and more histones, the histones get put together and the whole thing gets put back into that big chromosome you saw earlier. And once something's packed into a chromosome, it takes a whole different process to get that DNA out and use it again. This is a great way to shut things down. Something called RNA interference. Again, you use a non-cutting CRISPR-Cas9 structure, deactivated the molecular scissors, and you can get it to prevent transcription, the taking of DNA into RNA. That will stop the expression of the protein because you've got no RNA to build it from. So if you look at here, you see the RNA polymerase. That is the molecule you need to get DNA transcribed into RNA. This CRISPR just blocks it so it can't get to the DNA. That whole gene is shut off. Another thing that's kind of fun is that you can mark specific genes or sequences and visualize them within a cell. So you just put like a little fluorescent protein or something like that attached to your CRISPR-Cas9, and then once it binds, you can look under a microscope, see these genes. That could be useful if you're wondering, does that gene actually exist in this particular organism or its DNA? So really great way to go in and flag things that otherwise would be really hard to find, which means that poor old Waldo now is not as hideable as he used to be. You just get a CRISPR that recognizes Waldo and find Waldo really fast. So life was better for him before they found CRISPRs that could bind to Waldo. Okay, other thing I mentioned before, you can target multiple genes simultaneously. And this is another potential, they haven't developed it yet, but it's a potentially powerful tool because it can treat what we call multigenic disorders. That means there's a whole bunch of genes that contribute to the disorder. Heart disease is one of them. Some of the mental disorders like bipolar are brought about by this multigenic function. So you've got some pretty difficult things that now we might have an angle to treat them. This picture just shows several CRISPR-Cas9 molecules binding to different places in the DNA. Okay, genome editing at large is one of the things that it does. It's very powerful. That means that genetic material can be added, removed, or altered. See, this goes through the process of clipping the DNA. Then you can insert that little green thing, insert a new gene, or delete a section of an old gene. You can also alter it by mutating it. If you guys remember the video, there was that part where it showed trying to join blunt ends and it's error prone, so you can wind up with a mutation that can shut off an entire gene. One of the reasons that this is so damn cool is that this generates the potential of doing almost endless things. You do genome editing, specific genome editing to regular human stem cells, and you can generate stem cells that now can protect us from one of the greatest things that works is protecting us from cancer. Or rather, sorry, treating cancer. So this is a targeted way to treat cancers without having to invent an entire protein of some sort that you have to discover and develop. This goes in fast. And they've actually used something like this. It's out there being used now. And it's amazing because if you want to treat somebody for multiple myeloma, you don't have to wait to get somebody else whose bone marrow matches yours. You can make your cells become anti-cancer cells and put them right back into you. No immune reaction against them, no long, difficult transplant process. So this is already being used, still working the bugs out of it, shown some great power. Another thing that you can treat with CRISPR-Cas9 is some birth disorders. It's a good example of one that they're working on, treating a type of muscular dystrophy. Duchenne's muscular dystrophy results from single little piece of a gene. You can see the one that's numbered 51. If that's in there, it messes up one of the major proteins you need in your muscle cells, and they'll start to lose proper muscle structure and incorporate a lot of fat into it. Wind up with progressive muscle deterioration, weakness, and people who have this rarely live even a quarter of a century. This just shows the editing. They basically went through with CRISPR-Cas9, cut out that portion 51, and then when it was all put together, that mutated section is no longer there. You get healthy muscles, healthy muscle cells. As I understand it, this is actually in the early stages of being tried in patients. This works. You could be looking at a cure for muscular dystrophy. Jerry Lewis is somewhere in heaven smiling at that one. There's some potential future uses, and some of them are hardly even future now. Rapid diagnostic systems. They actually were engineering some of these during the current pandemic. And they are indeed very rapid. It's like, some of them are like 15 minutes from taking a sample from you to having a result in this. So incredibly fast. Another thing they can do is modify genes and pathogens so that they're less infectious. I mentioned that up with the the malarial mosquitoes. That's modifying the mosquitoes, but you can also modify the actual pathogens themselves. And again, modifying them directly in a way. This is really cool. Gene therapy can be done by modifying cells In this case, what that is up there is the sort of glass thing on the left is a system for holding single cells. Little bit that you see coming to penetrate the cell, that's injecting what you want, like this CRISPR-Cas9, into a single cell, and in this case, it's an egg, a fertilized egg. So you can take fertilized egg, a single one, modify it the way you want, and then put it into, say, an embryo, and get a specific type of, in this case, mouse you want. This thing could be used to research a specific gene. You can do the same thing with just cells or cell lines. Some more about the potential of CRISPR. There are about 30 to 40,000 genes in the human genome. Every one of those little colored slits in each of those chromosomes represents at least one major gene region. There aren't 3,000 bands in there, so you can tell there's a lot of material in there. And CRISPR, all the mutations that could exist in any of those genes could be targeted by the properly engineered CRISPR-Cas9 system, CRISPR-Cas12a. All right, that's kind of it. I just went through it. It's kind of quick, and at the end, it was mostly applications and just what its potential is like. I've got a kind of amusing video to end here, but before I click it on, I'd like some of you to throw questions out. We'll have this for entertainment at the end. So what questions do we have?
I have
Okay,
three.
yeah, go ahead. If you'd like to unmute yourself, you should be able to do that. And I shut down everybody's video just because I thought that would help the bandwidth issue, but feel free to open up your video as well.
Okay, I have three questions. The first one is, the PAM, is that one specific sequence so that you can only match things that start with that small sequence? Okay, go ahead.
Yeah, the PAMs, they do have a little bit of possible variation in them, but what they are is they're little triplet sequences that you can engineer, and you'll find them all over the genomes of bacteria, humans, whatever you're looking for. And they will grab hold of that little 3 nucleotide sequence that matches it. And then what it'll do is it'll sort of lay out the rest of the RNA and see if it makes a perfect match to that stretch of DNA. If it doesn't, then the PAM sequence will let go, whole thing will curl back up into itself, CRISPR-Cas9 molecule will go look elsewhere. So the PAM gives it a place to grab hold of, then it can check from there. If it doesn't match, it just lets go and goes elsewhere. This may sound kind of inefficient, but you have to understand that things that happen at the DNA molecular level, they can be happening at thousands of processes per second. So, you know, once you get used to the world of molecular biology, it's pretty freaky sometimes. Hope that answered your question well enough, did it?
Yeah, close enough. The second question is, it seems like the length of the RNA that you can match is limited to 20 or 21. Is that just because it all has to fit inside the Cas molecule? Too long, it wouldn't, that the whole thing wouldn't function correctly?
Yeah, you're right. If you made the thing, 80 bases long, it wouldn't fit. But the other thing is that when you're looking for a stretch of DNA that matches 20 to 40 base pairs, you're looking for something unique by the time it gets that long. You're not going to find a 20 or 40 base sequence all over the genome. You're going to find something like that pretty much in only one place. So 20 to 40 nucleotides is used because that's what you need. You don't need. It's also how the molecule has been designed in the bacteria. Bacteria have used it for millions of years and it works well for them, so we can use it too.
So it's good enough. Okay, and my final question is, for this to work properly in the bacterium, there has to be like hundreds of these things floating around inside the bacterium continuously looking for the bad DNA. Is that accurate?
To an extent, yes. They are floating around in that bacteria. They are looking for the viral DNA, but don't need a whole bunch of them. Just need a few. And then if one of them triggers, once it triggers, then that part of the DNA I showed you that encodes the Cas molecule and the CRISPR spacers and repeats, you can go to that particular section, turn it on, and then you'll produce bucket loads of CRISPR-Cas9 that are now looking for that specific viral sequence. So it's kind of like the Army scouts went out, found the enemy, called back to headquarters, and headquarters sent, massive amounts of infantry. So that's how that memory system works. It makes it faster next time it's infected.
Okay, so you might end up with a bunch of copies of it in the CRISPR to increase the chances that that one is being sequenced again?
Yeah, you actually will. Where you saw the spacers inserted,
Yeah.
the bacteria can basically insert about as many of those spacers as it wants. And then, like I said, all of those can be floating around in the bacteria looking for their matching sequence so that if they find it, they can pretty much tell that piece of the bacterial DNA to crank up production.
So you'd at least need to have one Cas9 that was instructed to look for each one of those sequences floating around at the same time.
Yes, exactly.
Okay, I'm done.
Okay.
Can you give us a little idea of what's going on with the coronavirus? Why it's so much tougher to crack than the others we've had over the decades?
Yeah, I did a coronavirus presentation a while ago, and I'm sure that's up on the site. But the thing that's making it tough to crack is that it's completely new. Nobody's ever seen this thing before, including all of us humans. That's why it's so deadly. It's also made up of sort of a chimeric joining of the DNA from two previously known coronavirus, which also makes it weak. It can mutate a bit, which makes it kind of a moving target. And the other thing is that in the history of humanity, we've never developed a preventative, a vaccine or something like that to deal with coronavirus. Because prior to this, coronaviruses have basically just caused mild infections that might give you a. So this is kind of like, we never had to target this before. It's completely new, and we're not even familiar with targeting this kind of virus. A lot to learn in a very short time. Okay.
And there's things that maybe we won't get lasting immunity once having it. Is that right too?
That is up in the air. Basically, because the coronavirus never really caused anything serious before, we didn't bother to investigate. Now, of course, we're having to investigate and determine whether or not we're generating those memory cells that I showed you earlier.
Right.
If we're generating those, then we're probably in good shape. There is some preliminary research that's very encouraging. It shows that for SARS, which is another coronavirus very similar to this, once you've been infected with it, you initially get antibodies, but those go away after a while, and what you wind up is memory T cell rather than B cell. T cells are the type of immune cell that are more likely to deal with viral infection. And they found that those T cells will persist as memory cells for up to 8 years. They don't have data beyond that because SARS was reasonably recent. So we may have lasting immunity. It just may be a little different than what we're thinking of with antibodies. Okay. That did it for you?
Yeah, Mike,
Yeah.
can the CRISPR-Cas9 be used to shut down this coronavirus in some way?
That's one of the methods they are indeed trying to use. They have CRISPR that will match some of the DNA sequences. That's what the rapid diagnostics are for. So they are trying to find, they are trying to find CRISPR sequences or create CRISPR sequences that can go in, find viral DNA, and chop it up. One of the things that makes it a challenge in that respect is we know CRISPR can do it. We've known that for a little while, but we haven't yet actually tried to generate one that's going to go into an infected person and deal with it within a short enough period of time to save their life. That's a whole new thing we've never tried before.
Yeah.
Been working towards it. Now all of a sudden we've got serious impetus to work on.
Does anybody have any other questions? I know, Chuck, it looks like you have another one, but before you ask your question, does anybody have any questions? Okay, Chuck, did you want to ask your question?
Yeah, well, I was going to comment again what you said. You know, me, maybe others, the physical science in the realm of simple to pretty complex books have been out there. And I've read over my not so short life just tons of stuff about the history of physics, and then more interesting, the current stuff that's going on in physics. And I think I can listen to it and have some understanding. But boy, this stuff on genetics is a subject that I've never gotten into. And it's really an interesting one. I hope to be able to continue my education. I just finished a book called Some Assembly Required. And this man gave a real good history. And again, of some of them in the last 100 years, he dropped names of scientists and the things that they've discovered in the last, whatever, that just, just something that has bypassed my education over the years. And I hope to find more that I can keep educating myself with.
Yeah, that's great. And, most folks don't have a whole bunch of biology background. Most folks don't have a whole bunch of physics background, chemistry backgrounds, or the science background. Pretty much got to be a scientist. And I know that I'm lacking quite a bit in some of the non-biological areas. And I'm also lacking quite a bit when you go into specific biology areas that aren't in my wheelhouse. So yeah, the world of biology is gigantic and fascinating to me.
I can understand that a lot of people feel like Chuck and don't understand the science behind this. So therefore, I see the future of this being people being very fearful of, okay, what is exactly going on? And because of the not understanding science really well, I'm not sure if this will be very accepted within society.
Yep, I agree. That happens a lot. And it's that classic thing of you fear the unknown. And you also misunderstand it and come up with silly things about it. To pick a different area of science, I thought this was hilarious. My ex-wife, her PhD in nuclear physics, she was mentioning that one time and somebody said, "How can you live with yourself doing that?" She had to scratch her head for a minute before she realized that this person heard nuclear and physics and thought nuclear bomb. Or questions or comments? Thing at the moment? Okay. to give you your little musical summary of what went on here. If any of you are interested, this comes from a site called Acapella Science. And this guy you see on there is a fantastic scientist. He's just brilliant. And he's also brilliant at making musical beats. I do want to ask one question before I start. This is about 6 or 7 minutes long. I was just going to play like one verse through of it. Does anybody want more than that, or shall we just do that short section? Let me know.
That's good.
What's that, George?
I was just saying that the short section was good.
I thought so too. Okay, here we go.
CRISPR-Cas9, bring me a gene, encoding for a specific protein. Make a few snips at this coded locus. You work so well inside a streptococcus. Cas9, I'm so alone without your scissors and my chromosome. Cut me up and do it clear. CRISPR-Cas9, bring me a gene.
Okay, that was where I was stopping, and I love that guy. Look him up sometime on YouTube, Acapella Science. He's got so much stuff, and it is hilarious.
That was marvelous. Thank you.
He's good. He's good.
Phenomenal.
If you like the space stuff, he did one. What's the one with Bruno Mars that everybody knows?
Happy?
No, Bruno Mars.
I'm not going to work today or something like that.
I want to be a millionaire.
No, the one everybody knows. It's kind of like the dance beat.
Uptown Funk.
What's that? Yeah, Uptown Funk.
Oh, yeah.
He took that and modified it to talk about the deep probe headed out to Pluto. He calls it Pluto. Yeah. Oh, yeah. He says it's written by Pluto Mars. But it's hilarious too. Okay, so that's what I had for y'all. Hope you enjoyed
Did you want to
it.
stop sharing your screen, Mike?
Yep, just got to find that. Where the heck did it?
At the top of your screen, I think there's a red box.
Oh, there it is. There we go. Sorry. Okay, got it.
Okay, people can share their videos if they'd like.
The video won't start. I think somebody turned
Yeah,
my video off.
Let me see.
I'm gonna be able to see my smiley face.
Yeah, I've asked you.
There it
I
is.
asked you to
Hey,
share
thank
your
you.
video. Here we go.
There he is. So, again, if anybody's still got questions or something pops into their head, let me know. Cheryl will take care of other details.
Says you still have mine stopped.
Same with mine, but it's not a big deal.
So I can ask you to start your video, but I can't turn it on. I turned it off, but I think you have the power to turn it back on.
Says you cannot start your video because now it's.
Or maybe I have to
Sorry.
ask you to
There we
start
go.
it first. Yeah.
Yeah, you do.
Okay. I have to give you permission to start it. Okay. I've learned something new. Awesome. So I had a question. Recently in the news, maybe a year ago, recently, there was a scientist from China, I think, that actually did some genome editing with CRISPR on
Babies.
babies, yeah. And then they were born. I think they were protective of the HIV virus. Do you know anything about that, Mike?
Uh, that specifically, I have not heard. I've seen other things that the Chinese have done that we can't do. They have very different regulations. Be shot for doing that in America. But yeah, the whole idea of treating somebody in the womb very early is part of how you would fix things like that Duchenne's muscular dystrophy. So you would need sort of your genetic testing early on while the woman is pregnant, and then based on that, you design a CRISPR system to try to basically fix the rest of the cells as the child grows. That's still a ways off for most ethical research, though, because it's kind of hard to go in and say, hey, Cheryl, can I treat your fetus and see if it comes out healthier? And you're looking at them going, what if it comes out dead? So it's still a ways off.
Isn't Down syndrome another candidate? Isn't it pretty much a gene or two problem?
It is a genetic problem, but it's not going to be treated by CRISPR because Down syndrome is an entire extra chromosome. So you think of one of those X's that I showed you with just huge amounts of DNA packed in. Down syndrome has an entire extra chromosome. The only way to treat it would be to go in, maybe in the womb, but how are you going to get rid of full extra chromosome from every single cell in the
Magic.
yeah, that's maybe
Any sufficiently advanced technology.
You need CRISPR-Cas9 Pac-Man. So when Cas9 recognizes that extra gene, it just attaches the Pac-Man to it, and it just keeps on munching till it gets to the end.
That would be fun, except it might eat the other two chromosome 21s, which would be another problem. I'm waiting for Star Trek technology so you can activate the transporter, beam them out, and then tell the transporter to grab one chromosome 21 out of every cell when it beams them back. So.
Tomorrow, not today.
Yeah, several tomorrows most likely. We've also got to figure out the warp engine.
So this is potentially a new governmental or global organization or agency that needs to be created to decide the use of genome editing for the entire world?
They'll probably go nation by nation and take suggestions from each other. There already are organizations that do this sort of thing. They all have different names. And off the top of my head, I'm just not thinking of any right now. Called bioethics is the whole field that studies that. There are bioethics committees at pretty much every university, at pretty much every sizable biological corporation, and then there are a number at the government level. So they're already there, and bioethics recommendations are usually followed. If they're not, they can be handed over to somebody like the NIH, the National Institutes of Health, and from there it'll actually be turned into a binding regulation or potentially a law if they really.
But again, to try to understand some of these things, I've heard that we have sequenced the genome of the human, but what that really means is interesting. And there's so many things on top of just the genes that makes them work or not work, whatever. That's the kind of stuff I got to try to find out in some kind of book that tell me about it.
When I mentioned the epigenomic stuff,
Right.
that is one of the methods that you're kind of talking about, chemically modifying the DNA so it either can be read or cannot be read. So that's just one of them. It's crazy. Parts of the genome will actually affect other parts of the genome. 30 to 40,000 genes doing this. You can wind up with whatever 30,000 combinatorial is. I have no idea what that number is, but it's huge. So, yeah, it's incredibly complicated. And I've only mentioned epigenomic processes. There are proteomic processes, chemical processes in and out of the cell, cells talking to each other. We'll be doing biology research for quite a long time yet.
We hear that the humans and the monkeys or whatever are 90 whatever high percentage the same genes, but then there's all this other stuff working on top that makes things different.
Yeah, it's those small differences that wind up making big differences. You know, the monkeys can't talk, we can talk, and otherwise we behave like monkeys. So not hard to believe we're very similar.
So this kit, can you buy this kit just on Amazon today and do experiments in your kitchen?
No, first off, you wouldn't have anywhere close to the amount of equipment and materials you need in your kitchen. There are things you can do in your kitchen. One of the kitchen tabletop things that's been used for a long time in high school biology classes is extracting DNA from things. It's actually a pretty simple process, and they usually use onions because the largest genome on planet Earth is the onion genome. I don't know why you need, 80 billion base pairs in order to just make layer after layer of the same thing, but that's what the onion is doing. So yeah, you can extract DNA on your tabletop. Doing much with it harder. And you cannot get the kits, like the diagnostic kits and stuff. Generally, you would not be able to order them yourself. You usually have to have some sort of a research designation. Frequently, the research projects you're on will have like an NIH designation. You can use that to order products.
But I'll bet the high school teachers doing a whole lot more with his high school kids than they did this a couple of years ago.
Yep. I enjoyed high school biology. I was particularly just completely in my element when I got to start cutting open and dissecting creatures. It's like, this is so fun.
So can this be used in non-productive or evil ways?
Oh, hell yeah. Oh, hell yeah. The one that just pops into my mind is if you create a relatively easily distributed form of CRISPR that is designed to cut critical genes in, say, us humans. So then you introduce it into, the bloodstream or whatever, and all of a sudden, all of my genes for producing tissue collagen are starting to fall apart. So am I. Just like physics, right? If you're a nuclear physicist, maybe you can make a bomb.
Right.
Yeah, that's why mad scientists are so popular in movies.
But again, that's back to Cas9, or the coronavirus. It's simply too complex for even the brilliant Chinese to have built and sent out in the world. Would you say that's probably true?
Now, actually, we could engineer a virus in that. It's possible. Many years ago, oh my goodness, this was in the 90s, some laboratory was doing research, and then without it really being known what they were doing, all of a sudden, poof, they came out with a complete paper outlining all their research. They had built a polio virus from scratch. They would just order short sequences of DNA, sequence other parts, keep building them, put those pieces together, and voila, they, like I say, de novo created polio virus. And everybody thought, well, theoretically, we think it can be done, but it must be too hard. These guys just proved it and scared the crap out of the whole biological community. The NIH went into an absolute tizzy. Oh, and by the way, on the coronavirus, you could have engineered it, but nobody did. If you do engineering to create something like that, basically wind up leaving things behind that other scientists can look at and go, oh, hey, this is artificially created. If it's a completely natural creation, see those marks.
And that's why I think the scientific community has said that the virus that's out there right now, you can tell that it's from natural sources.
Exactly. Exactly.
That doesn't mean that it was a natural virus that maybe accidentally escaped a lab or something. That's not talking about that. It's just talking about it's a natural source virus.
Yes, you could conceivably genetically engineer a coronavirus, but once you release it into the wild, you just lost all control of the dang. So who knows what it'll do next. One of these days, maybe we'll figure that one out, and then again, we'll probably scare the crap out of the entire biology community.
Can you go into, if you know about this, go into, you talked about the origins of the creation of the CRISPR-Cas9. There were two people, I thought, that I heard created it. One created the technology and one created the actual system to deliver that technology or something like that. Do you know anything about that?
Yes, you are correct. The ones I'm familiar with are Dudna and Chapartnier. Yeah, his name is hard to say. Chapartnier, I think is how he says it.
Yeah.
They are a pair of collaborators, and they have done a lot of the work on understanding this. I cannot remember the name of the engineers who put together a lot of the delivery. You're right.
Are they arguing or fighting over any sort of rights or patent rights to this?
Not as far as I know, but I can guarantee you that there are parts of it that if they're not being fought over, they will be fought over. We've had people try to establish patents on DNA sequences, which to me seems absolutely absurd, but I don't know if they succeeded or not. I have to ask my friend Mike Moran if they have. He's a biology lawyer.
But in trying to come up with a vaccine, I hear we're worried that the Chinese are, if we can get our research, why couldn't the whole wide world get together and say, let's find a good vaccine? We don't need to fight among ourselves in making the vaccine, do we?
No, and there is, there's kind of both. There's a lot of cooperation going on. Just to give you one idea, in normal situations, all the various scientific journals, some can be open access, a lot of them are not. Most of the scientific journals during this thing basically said, if it's a if it's a coronavirus related paper, it just instantly goes on open access. So the whole world, and there's a lot of, getting on the telephone and sharing knowledge. There are people actually physically shipping stuff back, and there are just unprecedented amounts of collaboration and information sharing with this thing. It's like nothing I've ever even imagined before. So, but there are people fighting and trying to be the first with a vaccine and crap like that.
Was
Any
one of
more?
those names that you just mentioned as the founder of Doudna? Was that?
Doudna or Dudna, however she says it.
Well,
Yeah.
okay. I have a book in my hand here, Kraken Creation, Jennifer Doudna and Samuel Steinberg. That might be a good book to read, I guess.
Yeah, it is. And another thing you could go look at if you like talks and video is Dudna has done a number of TED Talks about this. So just another thing you could look at.
Sure. Okay.
You could also look at the entirety of that video I kept showing you pieces of.
What's the name of that video?
If you go just look up CRISPR-Cas9 in YouTube, there's generally like 2 or 3 videos that'll come up on the top of the search every time. And I remember seeing a couple of them. They're both very good. So go in there and search. Look, if you really want that one in particular, look for a video that has pictures that look like the ones I showed you, because the others have different colors and models.
Well, thank you, Mike, for your presentation.
Thank you very
and
much.
I appreciate you researching that very quickly. I had asked Mike to change his plans on what he was going to present, and I thought this was a little bit more for the current days. But in the future, I think, Mike, you will still present your presentation on GMOs.
Yep.
Which has something to do with DNA.
Oh, yeah.
And so we'll put that on the schedule coming up in the future.
Great.
Anytime. That one's actually all done, because I expected to do it before Cheryl said, change.
See, I'm testing you. I'm testing you to see how quickly you can pivot and do some research.
Very fast. I'm going to brag. When I was working for the medical education company, they came in one time and they said, we have a collection of projects. There's about 6 of them. And there's going to be 3 other companies that we have to coordinate with. And the people that want us to do it have to have it ready for a conference that's happening in 4 months. Like, you want me to go from 0 to the speed of light in like half a month and then write the rest of it for 3 months? So I literally would be in the office until 1 in the morning. 2 nights, I remember the owner of the company gave me the keys to a nearby hotel room that we use for guests when they came to town. Because I was like, I'm not going to get out of here till 3 in the morning. I got to be back at 8. I can't even go home. So she said, here, go sleep here. But the stuff we produced kicked ass. Everybody who saw it at the conference was like, this is freaking cool.
Well, even your slide presentation, the way you've animated how things kind of slowly show up and all that, the layering of your pictures, I think is really cool.
Thank you.
Also, if you just want to bring in all that frickin' cool stuff and lay that on us, we'll watch it.
Here's my presentation. Wake me up when it's over.
That'll work.
Yep. A lot easier for me.
So Roger, I saw you came in kind of later than everybody else. It's nice to meet you. I don't think I've seen you before. Have you attended before?
He's videoed out.
Yeah, can you hear me?
Yes.
Yes.
Yeah, I haven't been in Boulder in a while. So, I'm currently in Georgia. I used to be a member when I was in Boulder.
Can you speak up just a little?
Yeah, this mic doesn't work very well. Is that better?
Okay. Yes.
Yeah.
Yes. So you're in Georgia right now, huh?
Yeah, I'm in Georgia.
Well, that's a good thing about Zoom presentations, Zoom meetings. We can get people from all over.
Yeah. It's an idea. If you want to do something big, you can contact another atheist group anywhere. Hey, we're doing this. Want to hop on?
Right. Well, speaking of collaborating with other secular groups, Thursday we have our community trivia, and we've invited the Jefferson Humanists, they're in Golden, and then the Secular Hub down in Denver, and then the Boulder Atheists. It's kind of a collaborative, fun event, trivia night. So you guys can join us Thursday night for that. Next Sunday, I don't have it on the schedule yet, but my son, George, is hosting that presentation, and it's called Answers in Genesis. And that's some books by Ken Ham. And he speaks to the, what do you call it, the 6,000-year-old Earth version and creationism. And so we're going to be delving into a lot of the logical fallacies from Ken Ham.
Don't forget the scientific outright lies.
Right.
So when are we doing that?
Next Sunday.
Next Sunday. Is it going to be an online thing like this?
Yes. Yes.
Cool.
So,
Right.
yes.
I have a picture of Ken Ham that I took in the lobby of the Creation Museum when I went and toured that. He
Oh,
was busy doing selfies with all of these families of good Christians that were coming in to educate their children. That was sort of sad.
Yeah. I actually met Kim Ham once way back when I was a Christian, and I went to a creationism seminar, and the guy is arrogant and unfriendly and concerned solely with himself.
really?
Big surprise. Yeah. Oh, and by the way, if any of you've seen pictures with him, he always has a beard. And so because of his craziness, some group that I was in used to just refer to him as the bearded loon.
Did not know that species existed.
It's rare and it's in Australia, like other weird things.
Yeah, I have some friends from Australia, and I just mentioned Ken casually in conversation with them, and they interrupted and said, on behalf of all of Australia, we would like to apologize for Ken Ham.
Oh, yeah, he's Australian, huh?
Yeah, they have a whole institute he created over there.
Wow.
Well, the reason my son George is interested in this is back in the day, one Christmas that we were celebrating with my side of the family, my brother and sister-in-law gave George these books, the Answers in Genesis series of books. And they gave them to him in response to the previous Christmas. An uncle had given George the book called Cosmos, Carl Sagan's Cosmos book.
Mm-hmm.
And they looked at that, and I think this was their response to giving George the other side of the story. So they are very religious still. And we brought those home and kind of looked through them, and I just couldn't really finish it. It's so icky in what it thinks is the truth. So he really wants to kind of present this and kind of point out the fallacies of it himself. So
Just one
It's
little
been years since
saw,
I read creation stuff.
I saw a little video of a debate, if you can call it that, between the creationists and the talking about the flood. And this more scientific man was talking about how much water it would take to cover the earth and the mountains and this creation. Well, you know, there were no mountains before the flood. I didn't know that.
Did not they
So-
land on Mount Ararat, which is
Yeah.
like 2,000 feet tall?
The first mountain climber as well as the survivor of the flood.
Mm-hmm. Oh, well.
Well, does anybody have any more questions? Otherwise, we can wrap this up and you guys can get on with your beautiful Sunday afternoon.
All right. Great presentation.
Thank you all. Very great.
Yeah, thank you, Mike.
Thank
Thank
you.
you.
Welcome. Glad you all liked it.
It was nice
And
to
I
see
will be-
everybody.
I will be, Cheryl, I got the one on GMOs, not GMOs, coronavirus. You got that up on our site somewhere?
I believe so. I will check. It should be, if that was being recorded, I'm not sure when we started recording the Zoom meetings, but I will check.
Okay, and I'm pretty sure I gave you the PowerPoint set at least.
Yes.
Okay, I'll give you this one too. Is that something, I guess I can email it. Hopefully it'll take something that big. Otherwise, oh, I know what I can do. I will put it on my my Google Drive and then share it to you, you know, send a link.
Okay. I think what happened with your viruses presentation is we put that on our blog.
Oh.
Can you hear me?
Yeah.
Yeah, we put your presentation, the virus presentation, on our blog. But since then, we've been recording these Zoom sessions and we're putting them on our website on another page to where people can just watch the whole Zoom recording. So
Okay. So you probably don't need the PowerPoint.
not for this, no.
Okay.
Okay, well, thank you, Mike, and good to see everybody. Hope to see you guys again. Thank you very much.
Thanks,
Right.
Cheryl.
Bye-bye.
Bye.
Bye.